Hey biology students, this is Mr. Davies, one of your biology teachers here at the high school. Today we're going to go through and we're going to be walking through one of our first flipped assignments that we're going to be doing on a virtual day. So there's certain days we're going to have these presentations that we're going to ask you to go through and fill out a note sheet. So you have a guided note sheet either linked in Canvas, or there were a couple paper copies available for you to pick up if you wanted to do that. As we go through this video, you're going to be filling in the missing words, the blanks, the definitions. It's a way for you to get information without actually being in front of us. So on these virtual days, we can still have you learning materials, so when you come back to class, we're ready to go. So that's the idea of that. When you're done, you can either be marking up the PDF and submitting that electronically via Canvas, or if you grabbed a paper copy, you can be filling in that paper copy and then submitting a picture and then turning that in Canvas. So you have some options on that, just so you're clear on that. So let's get started. So this is the first part of our biology unit, is going to be talking about, well, how do we actually determine things in biology or in science, and what are experiments, and what's a good experiment and what's a valid experiment. So the things that we'll be able to do when we're done is we'll know how to plan a scientific experiment, not just a random experiment, but a scientific experiment. We're going to know the differences between independent, dependent, and control variables, and we're going to be able to know how to graph data appropriately. So some things to keep in mind when we talk about our graphing. Our x-axis is on the horizontal plane, and our y-axis is on the vertical plane. So later on, we'll be talking about what goes on the x-axis and the y-axis. Remember, x is horizontal or flat, where y is our vertical or up and down. So some things again to keep in mind. So when we talk about variables in science, we have to have a hypothesis, and a hypothesis is something that we can test, and that's important in terms of having the validity of the hypothesis, is to make sure that is our hypothesis correct or incorrect. But in order to do that, we have to have certain things set up appropriately. Okay, so we have to make sure when scientists go through that they are planning their experiment, they are experimenting on only one thing at a time, and that is going to be the key. And I kind of skipped a step on you, and I apologize, but we only want to change one thing at a time. So we want to try to identify as many factors as possible that could cause a change or an observation, and the idea is we call that term a variable. And those are the things that are either changed or measured in a given experiment. So when we go through and we talk about a variable, we want to only have one variable change at a time. Okay, we only want to change one thing to see which variable is the actual cause of that change in the experiment. Okay, we don't want to change 6 things at once. We want to change one thing at a time. If something changes, then we can say, well, what caused that change? It was the one thing that we changed, which caused the change in the outcome. Okay, so there's a couple different types of variables, and these are just things you are going to need to know. There are controlled variables, there's independent variables, and there's dependent variables. So when we go through and we talk about what is an independent variable, what's the dependent variable, those are things we expect you to be able to know. It's what is the thing that you changed compared to what caused the change. The control variables are what we kept the same. But when we graph things, because in science we graph stuff a lot, when we talk about graphing stuff, again, our x-axis versus our y-axis. So when they talk about the independent variable goes on the x-axis, this is your x-axis. When they talk about on the y-axis, this is your y-axis. So the different types, the variable that we keep the same or constant is called our controlled variable. We want to try to have as many controlled variables or constants in a given experiment as we possibly can. The independent variable is the thing that is changed in the experiment. It's you as the experimenter, what you are manipulating. What are you changing? What are you going to, what are you trying to see, right? That's the idea. And then your dependent variable is what is going to be measured or what results in the change. So if we go through a given experiment with plants, we are going to try to determine what brand of fertilizer or is the best for growing for plant growth, right? So things that we're going to keep constant controlled variables are the amount of water they get, the type of soil they get, the amount of sunlight they get, and we're going to use the same brand of daisy seeds. We're going to use the same number of daisy seeds. We're going to use the same age of daisy seeds. We want to keep as many things as constant as possible. So our independent variable is the thing that we changed. The independent variable is, are we going to use Miracle-Gro? Are we going to use, and I don't know how to pronounce this, the Joby sticks or what? So the idea is, or are we going to use some different name brand? So our dependent variable is we're going to measure the thing that results of the change. We are going to measure plant growth. Okay, and that's going to be the thing that we're going to measure. So does Miracle-Gro make our plants grow more? Do these sticks allow it or cause it to grow more? So experimental design is something you've done many times. And it says team up with your pod and write this down. We aren't going to be in pods. We're doing this virtually, but the idea is the same. Think about something you've seen in your life that doesn't have to be science related. You're going to take some observations and create a hypothesis. Okay, so a hypothesis is often as an if-then, and then come up with ideas of how you would test this. So the idea could be if I drop a crumpled piece of paper up, is it going to fall faster or slower than an uncrumpled piece of paper? Okay, that's the idea. So if that was yours, so take a second, kind of think about this, and then talk yourself through the steps. Is what's your independent variable? What did I change? Well, in my situation, crumpled paper versus uncrumpled paper. What's the dependent variable? How long it takes to fall to the ground. What are the controls? Well, I'm going to want to use the same type of paper, the same size of piece of paper, the same color of piece of paper. I'm going to drop it from the same height. It's going to be in the same classroom with the same amount of wind. As many things as possible. And something that we want to make sure we talk about is sample size. We don't just want to drop something once and be done. We want to do it multiple times to get multiple points of data. So it's just not was that a fluke occurrence or a random occurrence that caused it? We want our results to be based upon repeatable data. And we have to keep in mind that there's going to be some errors. And that's something that we need to identify, and we try to say what caused the errors. Don't just say, well, there was human error. I don't know what human error is. Was the human error like I wasn't very precise with my stopwatch time, start and stop time? I was delayed. I was trying to drop it and start my stopwatch at the same time. Well, that's a human error. So what can we do to improve that? Well, we could get someone else to time it, something like that. So we have to be aware, and that can make actually an experiment invalid. And if we have an invalid experiment, the results of that experiment are not valid, so we can't use them. We can't say, hey, I did this experiment. It wouldn't really work. It was flawed, but we should still talk about it because it's cool, or we should change what we do as a practice of human society based upon this experiment, even though it was invalid. If it's invalid, we can't use that data, and it shouldn't be talked about, or I guess not talked about, but it shouldn't be pushed out there as science because it was invalid, therefore it's not a scientific experiment. So what makes something invalid is if you have more than one variable. We cannot change more than one independent variable. If you change more than one, you don't know what caused the change. If I change the height of my crumpled piece of paper, if that's up higher than my non-crumpled piece of paper, well, I changed the shape of the paper, crumpled versus uncrumpled, and I changed the height. So why did the crumpled piece of paper that was closer to the ground hit the ground first? Was it because it was crumpled, or was it because it was closer to the ground? The answer is we don't know. Therefore, we can't draw a conclusion from that because it's not a valid experiment. Okay? And then we have to go through and identify what you should have a group that is not manipulated, and then that's the idea of our control group. That's the thing we're going to compare it to. So in that case, the crumpled versus uncrumpled, the uncrumpled would be our controlled group, and that's what we are comparing it to. So we need to have a control group, otherwise we just have randomness and we don't have anything to compare it to. So when we graph things, important things that we need to make sure we include in graphs is we need to have a title. I apologize, that color did not show up very well. Let's try the pink. We need to have a title, and it is usually written as a variable, y variable versus your x variable. So what's on your y versus what's on your x is often. So this would be like number of E. coli bacteria versus hours of incubation. We need to make sure we have labels that include units. When we go through, we plot and we do a best fit line. We don't play connect the dots. We don't go like boop, 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 right? We do a smooth fit, the best fit. And the idea is we get some randomness in our results that aren't going to be precise. And we need to account for that. And we want to draw a conclusion, a mathematical conclusion, which is why we do a best fit line. When we make a graph, the idea of the entire page is to make sure we can see our results. You don't want a graph that had like this big graph that does like this, right? And you maybe there like that. You want it to use your entire grid, your entire graph, because then we can see what's happening with your data. What might look like a straight line is really a curved line when we scale it appropriately. And we need to have our increments even. So notice like here, these increments are the same space apart. We have the same scale. We don't notice this scale and this scale are different. These go by 20s, these go by 1s, but the scale on the x-axis is constant and the scale on the y-axis is constant. And if we need to provide a key. Okay, so that is the first lesson. You should have, as you've gone through, filled out the guided notes that went along with this. If you have any questions, make sure you are ready to ask those questions when you are in class on your next in-person day. Have a great day, everyone.